Welcome back, everyone. Today we're going to talk about enzymes and just kind of the basic structure and function of them. And then in class, in person, we'll get into ways that we can speed up or slow down their effectiveness. So let's dive in. Here are the key terms. And you've probably heard of enzymes before, but you might not completely understand what they are. They are proteins, so they're a class of proteins, and their whole job is to speed up chemical reactions. Another way that we say speeding up a chemical reaction is to catalyze it. So enzymes catalyze reactions or speed up reactions. Now, the way we name enzymes in biology kind of helps us identify them a little bit better. So if a compound name ends in ase, like catalase or polymerase or anything like that, alcohol dehydrogenase, anything that ends in ase is an enzyme. Now, there are some that don't fall into that category. However, for the most part, you can pretty much rely on ase equals enzyme. Now, we might think, okay, that's cool, it helps us speed up reactions, but I don't think we really fully appreciate how important enzymes are. Most of the reactions required every single second for life to continue, if they were allowed to happen randomly, on their own, in their own time, it could take over 1,000 years for some of these reactions to occur. Because every single reaction that occurs in a cell is the result of random molecules bumping into each other. Not only do the molecules have to bump into each other to chemically react, but they have to bump into each other at just the right angle where the reaction can occur between the bonds that it's going to occur between, and with the correct amount of speed or velocity to actually get a reaction to occur. So you might have the two molecules that need to react. You might have them bumping into each other all the time, but they might not be bumping into each other with enough force or in the correct orientation. So enzymes are incredibly important because they take away that burden of needing to be just right with just the amount of energy. They actually facilitate the reaction to occur even if the molecules aren't colliding exactly perfectly. So without enzymes, most chemical reactions would never occur. And so as a result, enzymes are essential to living things. We have them in every single one of our cells throughout our bodies. They are responsible for pretty much almost every reaction occurring all the time in our cells, that is. So how do they work? Every single chemical reaction requires some energy to get started because in some way or another, you're either breaking a bond or there's some energy toll that has to be paid for the reaction to occur. Sometimes this is a large amount of energy that has to be put in. Sometimes it's pretty small and it's easily overcome. But there is some amount of energy that has to be paid before every reaction can occur. And so, as a result, enzymes step in and lower that amount of energy. We call it the activation energy. So without an enzyme, we might have a high amount of energy, the activation energy that the reactants have to overcome for the reaction to occur. With the enzyme, we lower that amount of activation energy needed. How do they do this? Well, they place the molecules in the proper position. So as I said, not only do the molecules have to interact with each other, they have to interact at just the right angle with just the right velocity. So in this case, the enzyme puts them into the correct place and also helps assist in the breakage of the bonds so that they can then create that chemical reaction. And usually the covalent bonds that enzymes are working on are highly, highly stable. So it does take quite a lot to get these bonds to break and to react. So that activation energy is the energy required to begin a chemical reaction. And enzymes work by lowering the amount of activation energy needed to start a particular reaction. So we have a high amount of activation energy normally, and enzymes with the reactants will lower that. Now, this is just an example graph. It might be significantly higher, it might be lower, this might be even further lower. It just depends on the enzyme and the particular reaction. So let's talk about how this actually occurs. Let's look at the structure of the enzyme and get into how that ends up creating the reactions. So enzymes can either take a compound and break it down into smaller pieces, or it can take multiple pieces and bring them together and form a larger molecule. So we're just going to look at one example of that in this picture here. What we have here in gray is the enzyme, and this green structure here is representing a compound that the enzyme is going to cause to chemically react. We call this compound that's going to enter into the enzyme the substrate. So any chemical or chemicals that the enzyme is going to act on is the substrate. Then the substrate fits into a very specific spot on the enzyme, and it's actually a physical fit. It's both a physical and a chemical fit. And we call that spot the active site. The active site is where the chemical reaction is going to occur. So it's the spot on the enzyme where the substrate fits in and the chemical reaction occurs. And then it creates products. And the products are just whatever is created at the end of this chemical reaction, either two different products from the original substrate, or this can also work in reverse. We can have two different substrates enter in, and the enzyme will bind them together into a product that is now larger and composed of the two pieces that originally came in. Now, this whole process is highly, highly specific. We say that the enzyme and its substrate act like a lock and a key. So each enzyme has one substrate that it will work on, meaning either one molecule that gets broken down or a set of molecules that get put together. So it's not necessarily that an enzyme can go and cause any reaction that it wants to happen. It is highly, highly specific. Only the specific substrate can fit in that given enzyme. Now, it's a little bit more complicated than that. It's not just that it fits in and it's a perfect fit like a lock and a key and we're all done. What I mean by lock and key is more the specificity. I can't go unlock your home door with my key. That's the idea there. But really, in real life, that's not a perfect analogy, because what happens is actually something called the induced fit model. So what happens is the substrate will enter into the enzyme in the active site, and it actually causes the enzyme to change its shape just a little bit. And when it does that, it actually causes the substrate to fit even better than before. So it kind of has to be able to enter in, and then the enzyme will hug it just right. So it would be like if you stick your key in the lock and the lock actually changes just a little bit to perfectly accommodate just your key, and it holds and hugs the key exactly tightly. Now, this is because usually of hydrogen bonding. So as the substrate comes in, it will form hydrogen bonds with the enzyme surrounding it, and that causes a slight what we call conformation or shape change. So this just causes the substrate to move into the exact proper position for the reaction to occur. Because remember, it's velocity and position when we talk about these chemical reactions occurring. So by putting the substrate in the exact right alignment, we don't need to use as much energy to do this reaction because everything's lining up perfectly. Now, enzymes are recyclable, they're reusable. They do this whole reaction and then can immediately do another one. We don't break them down, they don't fall apart after a reaction. We don't have to build it up again after it's done. They're kind of like Pac-Man. Pac-Man can go and eat a little dot and then immediately eat another dot and then another dot. It can just keep going. So they are not changed by the reactions that they catalyze. So this means we can reuse the same enzyme hundreds to thousands of times. And this is really, really important when we consider that life is always worried about where it's going to get its energy from. And energy is a finite resource to living things. It's very difficult to acquire. So we don't want to do things that waste energy. It would be highly wasteful of the cell's time and energy to constantly break the enzyme down and reforge it. So instead, we have the enzyme stay as it is so it can continually do multiple reactions. Now, eventually, the enzyme will be broken down for parts and remade because all molecules eventually wear down and kind of have natural wear and tear on them from all the collisions that they're experiencing in the cell. But sometimes we also want to pull back on how many enzymes we have to slow down our reactions. Sometimes we want to make more enzymes to make a reaction happen more frequently. So there's obviously an asterisk here. However, it's best to just make an enzyme and let it work as much as it can. Now, we can have extremely fine-tuned control over our enzymes. We can control when we make them, how many we make, when we want them to break down, all of this, but we can also control them as they're existing inside the cell or inside the body. And we do this with activators and inhibitors. So activators are chemicals that will bind a secondary site, so not the active site. They'll bind somewhere else. We often call that an allosteric site. So they'll bind somewhere else on the enzyme, and when that happens, it causes a slight shift in the structure, a slight conformational change in the structure of the enzyme to make it work better. So often these activators are things like our vitamins or ions and electrolytes like magnesium and calcium. So those are our activators. This makes an enzyme work better. On the opposite side of that, we have inhibitors, where these are molecules that actually stop or inhibit or reduce the enzyme's ability to function. Now, inhibition can be permanent, so an inhibitor can bind to an enzyme and never come off, or it can be reversible. And the body tends to, and the cell tends to be very specific with that. Either it will create an inhibitor for a period of time and then allow it to fall off, or sometimes there's toxins in the world that want to permanently inhibit something. So it just depends on the situation. Now, there's two ways that an enzyme can be inhibited. The first one is something called competitive inhibition. So this will occur in the active site. So we have the active site here. It looks like a little slice of pizza got taken out. And then this round site that looks like someone bit into it, this is the allosteric site or a secondary site on the enzyme. So with competitive inhibition, the substrate and the inhibitor compete for the active site. That's why it's competitive. They're competing over the same spot. So if the substrate goes in and fits in the enzyme, the reaction occurs. If the inhibitor gets there first and binds to the active site of the enzyme, where is the substrate going to fit? It can't fit in the active site, so no reaction is going to occur. The second way we can inhibit an enzyme is through non-competitive inhibition. So this is where the inhibitor doesn't fight with the substrate over the active site. Instead, it's going to bond to that secondary or allosteric site. So it's going to bind there. And just like with our activators, when something binds to the enzyme in a secondary site, it causes the enzyme to slightly change its shape or its conformation. In this particular case, it does not make the enzyme work better. What will happen is it will actually cause the shape of the enzyme's active site to be different. As a result, the substrate can no longer fit inside the active site because its shape has changed as a result of this binding to the substrate. So when the substrate tries to fit in, it can, or I'm sorry, the inhibitor here, not the substrate. So when the substrate tries to fit in, it can't, and no reaction can occur. So competitive inhibition fights over the active site. For non-competitive inhibition, the inhibitor goes to an allosteric or secondary site, causes a shape change in the active site so the substrate can no longer fit. And that is it for enzymes. Let me know if you have any questions, and I will see you in class.